The DNA code. The double helix of the DNA molecule is what we see here. We have two strands of DNA. One is the antisense strand, and it runs from 3' to 5', which means the deoxyribose sugar on the first nucleotide has a free 3' end, and at the end of the strand, the deoxyribose sugar is bound at its 5' end to a phosphate group. And in opposite, we have the sense strand, which runs from 5' to 3'. So in transcription, which happens in the nucleus, the enzyme RNA polymerase unwinds the two DNA strands, and on the template strand, which is the antisense strand, we have a promoter region. This is where the RNA polymerase recognizes that we have a gene. After the promoter region, we directly have the coding region, which is the code for the actual protein. So this is where transcription actually starts. In the coding region, we will start to build an mRNA strand complementary to the template DNA strand or the antisense strand. Why we use the antisense strand is because the RNA polymerase reads DNA from 3' to 5' direction. So this keeps happening until we reach the termination sequence, which is the sequence of DNA after the coding sequence that tells the RNA polymerase to stop transcription, and the mRNA leaves and goes into the cytoplasm to undergo translation. Now in translation, the mRNA will bind to the ribosome to be translated. Translation happens by the triplet code. We have every 3 nitrogenous bases are going to code for 1 amino acid. UGG codes for tryptophan. This table shows us how to look at the DNA code. We see codons, they show us which amino acid they code for. So for example, let's take UGG, which is the first codon we see here. We look at U on the left. This is the first nucleotide. The second nucleotide has a nitrogenous base G, so we look at G up in the region of U, and then we go to G, which is the third nitrogenous base, UGG, we found it. It codes for TRP, which is tryptophan. Now the second codon is UUU, so we go to U, U, and U. We found it. It codes for PHE, which is the amino acid phenylalanine. The third one is GGC. It codes for glycine, as we see in the table, and finally we have UCA, which codes for serine. So this is part of the polypeptide chain. Every 2 amino acids are bound together by a peptide bond, and when translation finishes, we have a polypeptide strand, which is going to make the protein. And this is how we transcribe and translate DNA into a protein. So we have 4 nitrogenous bases in mRNA, A, U, C, and G, and every codon is 3 bases. So 4 to the power 3, we have 64 possible codons, but we only have 20 amino acids. So we have 64 codons coding for 20 amino acids. This means that more than one codon are going to code for the same amino acid. Look at the table here, for example. Take leucine, LEU. It has 4 codons that code for it. And this is very important. We're going to discuss it later on. Also, the triplets of bases that code for individual amino acids are the same in all organisms. So UGG codes for tryptophan in humans, bacteria, bananas, monkfish, or any other organism you can think of. And that's why we say the DNA code is universal. Not only is it universal, but the DNA in different organisms is very similar. We share 98% of our DNA with chimpanzees and half of our DNA with bananas. So this shows how important DNA is as a universal code.